What exactly is DNA replication? DNA replication is the biological process in which one DNA molecule is copied to produce two identical DNA molecules. Yes? This process happens during the S phase. Yes? The S phase. Or synthesis phase. Yes? Synthesis phase of the cell cycle. The letter S stands for synthesis because this is the stage when new DNA is synthesized. Replication always occurs before the cell enters mitosis or meiosis. If DNA were not copied first, each daughter cell would receive only half of the genetic information, making survival impossible. Yes? So, one of the most important concepts in AP biology is understanding that DNA replication is semi-conservative. But what does that actually mean? Yes? I will explain for you. Scientists once proposed 3 different models to explain how DNA might replication. The first was the conservative model. Yes? Conservative model. In this model, the original DNA molecule would remain completely unchanged, while an entirely new DNA molecule would be produced. Yes? Like this picture. The second possibility was the dispersive model. Yes? Dispersive model. According to this idea, every strand of DNA would contain a random mixture of old DNA and newly synthesized DNA. Yes? Finally, scientists proposed the semi-conservative model. In this model, the 2 strands of the original DNA molecule separate from one another. Each original strand then serves as a template for building a new complementary strand. Yes? As a result, every newly formed DNA molecule contains one old strand and one newly synthesized strand. Later, the famous Mendelssohn-Stahl experiment confirmed that the semi-conservative model was correct. Yes? Semi-conservative. Yes? Confirmed. Even today, this remains one of the classic experiments you should know for AP biology. Yes? Now that we understand what replication is, let's review the structure of DNA itself. Understanding DNA structure makes replication much easier to understand. Yeah, guys? DNA has the shape of a twisted ladder known as a double helix. Yes? Double helix. The sides of the ladder are made from alternating sugar molecules. Yes? Sugar molecules and phosphate groups. Yes? Together they form the sugar phosphate backbone, which provides strength and stability to DNA molecules. So, I will summarize for you. Sugar phosphate backbone provides what? Provides strengths and stability to the DNA molecule. Got it, guys? The steps of the ladder are made of 4 nitrogen-containing bases. Yes, these bases is adenine, thymine, cytosine, and guanine. Yes, these bases do not pair randomly. Instead, DNA follows very strict complementary base pairing rules. Yes, adenine always pairs with thymine with two hydrogen bonding there's yes 2, and cytosine always paired with guanine. Yes, with 3 hydrogen binding. Yes, these specific pairing are held together by hydrogen bonds. Adenine and thymine share 2 hydrogen bonds, yes, I said you, and cytosine and guanine share 3 hydrogen bonds, making this pair slightly stronger. Because of these pairing rules, if you know the sequence on one DNA strand, you can always determine the sequence on the opposite strand. Like what? Like ATTGCA. We can write the sequence of the opposite strand TAACGT. Yes? So, another key concept for AP biology is that DNA strands are anti-parallel. Yes? Anti-parallel. Yes? These words may sound complicated, but the idea is actually simple. Each DNA strand has a direction. One strand runs from the 5' yes, end to the 3' end. Yes? Because the 2 strands run in opposite direction, they are called antiparallel. Yes? This arrangement becomes extremely important because DNA polymerase, the DNA enzyme responsible for building new DNA, can only add nucleotides in one direction. Yes, DNA polymerase. Yes, this single rule explains why one strand is copied continuously while the other must be copied in small pieces. We'll explore that in the next part. Before moving on, let's quickly summarize the key ideas we've learned. Yes, I will summarize this video for you. So, number 1. DNA replication occurs before the cell division during the S phase of the cell cycle. Yes, DNA replication in S phase. Is it true, guys? Before mitosis. Yes? Number 2. Replication follows the semi-conservative model. Yes? Semi-conservative model. Meaning, what's meaning semi-conservative? Can you say? Meaning each new DNA molecule contains one original strand, yes, and one newly synthesized strand. Yes. Number 3. DNA is made of 2 antiparallel strands arranged in a double helix. Yes. 2 parallel strands arranged in a double helix. Yes? Complementary base pairing ensures accurate coupling with adenine pairing with thymine and cytosine pairing guanine. Yes? Understanding these concepts provides the foundation for everything that happens during DNA replication. Yes, guys? In the next part, we'll follow the very first steps of replication as a specialized enzyme locate the origin of a replication, unwind the DNA double helix, and prepare the molecule to be copied. Have a good time.